In this video, we'll cover the first stage of photosynthesis, which is the light-dependent reactions. Let's first review the overall function of the light-dependent reactions, which is to transform light energy into the chemical energy of NADPH and ATP. This chemical energy will then be used during the second stage of photosynthesis, the Calvin cycle, to build a carbohydrate. We're going to start with the photosystems. What are these? Well, they're huge protein complexes embedded in the thylakoid membrane. They have multiple proteins that are bound to pigment molecules, the most important of which is chlorophyll A. There are two photosystems, photosystem 2 and photosystem 1. We're going to start with photosystem 2, which might sound a little backwards, but here's the thing. They were named in the order they were described. So photosystem 1 was discovered first. They probably just called it photosystem. And then when photosystem 2 was discovered, they called it photosystem 2. Also, kind of coincidentally, that's the order that they evolved in. So that's kind of, something you can think about in terms of their names. But in terms of our normal process of the light-dependent reactions, we're going to start with photosystem 2. All right, so now let's add light. Photons of light are absorbed by the pigment molecules, which become excited. Excited means that the electrons of the pigment molecule have been lifted to a higher energy level. This is a very transitory state. The electrons will not stay excited for very long because the situation is unstable. So they will, quote, fall back to their normal energy level, and the excess energy will be released in the form of light, a photon, and heat. The light photon that is emitted then excites a nearby pigment molecule, and the same process happens again. Electrons of the pigment molecule are excited, they're lifted to a higher energy level, and then as they return to their normal lower energy level, a photon as well as heat energy are released. This process continues. Energy is passed from pigment molecule to pigment molecule until the reaction center is excited. The reaction center, when we talk about that, we're really talking about two chlorophyll A molecules that absorb photons at a wavelength of 680 nanometers. So we call it P680. At this point, an electron in P680 is excited enough that it's actually released from the molecule and accepted by another molecule, labeled here as the primary electron acceptor. The excited electron is actually passed to another protein embedded in the thylakoid membrane, which is plastoquinone. So we've got a redox reaction on our hands here, right? P680 has lost an electron, so it's been oxidized, and plastoquinone that accepted the electron has therefore been reduced. Electrons that leave photosystem 2 need to be replaced, and that takes place when P680 actually strips electrons away from water. So when P680 grabs these electrons, it's reduced back to its normal state, and water is oxidized. And without those electrons, it becomes oxygen and hydrogen ions. We write this as 1/2 O2 because each water molecule only has one oxygen atom to release. This oxygen atom immediately binds to another oxygen atom that's been released by the oxidation of water to form O2, or oxygen gas. Okay, now let's follow the electron that's been accepted by plastoquinone. And hey, look at this. There's an electron transport chain. Electrons are passed from plastoquinone to cytochrome B6F to plastocyanin. And two of these proteins can actually pump hydrogen ions against their gradient from the stroma of the chloroplast to the thylakoid lumen. This is active transport because we're moving something against its gradient. And why do you think this is happening? Well, you remember chemiosmosis. The hydrogen ion gradient is used to power ATP synthesis. Hydrogen ions diffuse down their gradient, releasing energy that's used to produce adenosine triphosphate from adenosine diphosphate and inorganic phosphate. This should look pretty familiar by now. Let's continue our journey through the light-dependent reactions now, and to do that we need to take a quick look at photosystem 1. Here it is, and it looks a lot like photosystem 2. The main difference that we're going to highlight is that the reaction center, which again is a pair of chlorophyll A molecules, absorbs photons at a wavelength of 700 nanometers, so we call it P700. But it functions similarly. Light energy is absorbed by a pigment molecule. Energy is passed from molecule to molecule until it reaches the reaction center. P700. Electrons from P700 can become excited enough that they leave P700. So P700 has been oxidized, and they are accepted by an electron acceptor and then passed to ferredoxin. The electrons that leave P700 are replaced, similar to P680 in photosystem 2. But here they're not replaced by electrons from water. Instead, they're replaced by electrons from plastocyanin. In other words, from the electron transport chain that began with electrons from photosystem 2. All right, let's check back in with the electrons that are passed to ferredoxin. They're passed by ferredoxin to NADP plus reductase, which finally passes them to NADP plus, which is thereby reduced to NADPH. And NADPH, just as a reminder, this is an electron carrier, extremely similar to NADH in cellular respiration. It's now energized and ready to deliver that energy to the molecules of the next stage of photosynthesis, which is the Calvin cycle. All right, so let's just check back in with our overall summary. We've captured light energy and transformed it into the energy of ATP and NADPH. Those are ready now to be used during the Calvin cycle. And we've also produced oxygen gas as a product of taking electrons from water and using them to reduce photosystem 2, or P680.